Hello, I'm SabahMusfiah from the Department of Electronics Convergence Engineering. Today, I will give you a short summary of a research paper titled Reagentless Aptamer based on the ultrasensitive and fast response electrochemical capacitive biosensor for EGFR detection in non-small cell lung cancer. So, this paper talks about a fast and sensitive biosensor used to detect EGFR, a protein linked to lung cancer. The sensor uses a special DNA strand called an aptamer, which is made by Select's process and fixed on a gold electrode. It can detect EGFR within 3 seconds and even at a very low concentration of 0.005 nanograms per milliliter. The sensor can be used up to 5 times and does not need extra chemicals. That's why it's called reagentless. Because it's quick, portable, and level free, this biosensor could be very helpful for early and easy lung cancer diagnosis. First, I'll give a short introduction. Lung cancer is one of the main causes of cancer deaths, and non-small cell lung cancer makes up about 85% of the cases. Early detection is very important to help patients survive. Next, EGFR is a key biomarker for non-small cell lung cancer. It's a protein that often mutates in cancer, and these mutations can be treated using special drugs called tyrosine kinase inhibitors. However, current detection methods like PCR or antibody tests are expensive, slow, and complex. That's why researchers use aptamers, which are short DNA or RNA strands. They are cheap, stable, and very specific, making them good for sensors. Capacitive biosensors detect changes in capacitance when molecules bind together. They are fast, sensitive, and easy to make small. In this study, the team developed a capacitor-based biosensor using an EGFR tamer. It can detect EGFR in just 3 seconds and is reusable, low cost, and effective for early lung cancer diagnosis. In this study, the researchers used different cell links like HCC827 and A431, which are grown under standard lab conditions. The aptamer used was a 53 nucleotide DNA strand, specially modified to make it active and stable. The biosensor was made with gold and chromium electrodes. These are interdigitated capacitor structures with 24 fingers and a PDMS layer for better stability. To prepare the electrode surface, they first added MPa for binding, then attached the aptamer, and finally used MCH to block unwanted reactions. They measured capacitance changes using an LCR meter and also checked the surface using tools like AFM, SEM, and FTIR. The team also performed binding and fluorescence tests to see how well the aptamer could detect EGFR. In animal tests, they checked EGFR detection in mice lungs. All results are statistically analyzed and only results with p less than 0.05 are considered significant. First, the team developed the IDCE biosensor, which is low cost, very sensitive, and fast. It was made using gold and chromium electrodes, and surface tests like SEM and AFM confirmed that the sensor was well structured. For the surface characteristics, they found different surface roughness values, around 7 nanometers for MPa, 29 for the aptamer, and 60 for MCH, showing that each layer was successfully formed. Next, for specificity and sensitivity, the sensor showed a linear response and 500 Hz. It could detect a very small amount of EGFR less than 0.005 nanograms per milliliter and responded within 3 seconds. When comparing aptamer and antibody, aptamer was much more sensitive and had less cross-reactivity. Also, cell line tests confirmed that the aptamer works well with EGFR expressing cells. In KRAS mutant mice, The aptamer showed a 210 times higher signal in the lungs compared to normal mice. This means the sensor can clearly detect cancer-related changes. The biosensor was also reusable up to 5 times but became unstable after 6 cycles. Tests confirmed that the aptamer remained stable and could be cleaned effectively. Overall, this biosensor is highly specific, very fast, and ultra-sensitive for EGFR detection in lung cancer. In the future, it could be improved by adding machine learning nanocomposite materials or used as a portable diagnostic device for early cancer detection. To summarize, this study developed a reagentless EGFR aptamer for early detection of non-small cell lung cancer. The aptamer was firmly attached to gold and chromium electrodes using MPA treatment. The sensor showed high specificity, meaning it can clearly tell EGFR apart from other proteins. It also has a very fast response with results appearing within 3 seconds, and it works best at 500 Hz. Overall, this biosensor is fast, low cost, and suitable for real-time cancer diagnosis. In the future, the researchers plan to improve the stability and selectivity, test it with real patient samples, and develop a portable version that can be used easily for continuous monitoring. That's it for today. Thank you.